Good morning, good afternoon, and good evening, everyone. And welcome to this Scientist.com Inside Scientific and Indigo Biosciences webinar titled How to Better Predict Potential Drug Interactions Early in the Discovery Process. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be the host. Today, we're being joined by Dr. Jack Vanden Heuvel, Professor of Molecular Toxicology at Penn State University and co-founder and CSO of Indigo Biosciences. In his presentation, Dr. Vanden Heuvel will present effective strategies for early detection of drug-drug interactions using in vitro assays and gene expression studies. And with that, I'd like to welcome Dr. Jack Vanden Heuvel. Jack, thanks so much for joining us today, and the floor is yours whenever you're ready.
Great. Thank you, Liam. And I'm excited to talk to you today about drug interactions and how this can be studied using relatively simple in vitro systems where you can save money, save time, and something that's done early in the drug discovery process. And how I'm going to do that is shown in this outline slide. So I'm going to start out talking about some basic pharmacokinetics, in particular absorption, distribution, metabolism, and excretion. And then I'll introduce the adaptive response, which is very important for the area of drug-drug interactions. And then I'll bring up some of the important ramifications of drug-drug interactions and some of the problems. And then I'll go into some examples and applications of looking at nuclear receptors in particular and how they're involved in drug-drug interactions. And at the end, I will summarize and go over some of the high points. So let's start with the basics of pharmacokinetics. And in order to understand how a chemical is going to be processed, looking at things like half-life and effective concentration, we have to look at things like ADME, or absorption, distribution, metabolism, and excretion. And when we're talking about this, we're going to be talking about xenobiotics. So these are any chemical that is foreign to that organism. So we tend to think of this as a drug, a nutrient, a pollutant, again, something that is not normally produced within our body. And the process, of course, starts with when we're talking about oral absorption of a drug, liberation. If it's a food component, it has to be dissolved, and then it's absorbed, goes into the central compartment, which in most cases we're thinking of the blood or the plasma. From there, it can go through a process of binding to proteins, can go into tissue reservoirs. It can partition into sites of action, both for on-target and off-target effects of that drug. And all of these together are considered the distribution, where it's going within that organism. The parent molecule or free xenobiotic can be metabolized. When we're talking about drug metabolism within an organism, this is biotransformation, where we have enzymes that are responsible for producing the metabolites. And then the parent molecule and any metabolites that are formed are then excreted, and we're seeing a decrease in the amount of the body burden of that particular xenobiotic. One thing that I want to point out that's very important for ADME is that many of these processes require your chemical to cross biological membranes. And this could be true for the absorption phase, going from the intestine into the bloodstream. It could be going from the bloodstream into the tissues where it's having its activity, into storage forms, and then also when it's going from the central compartment into the excretory organs, it also has to cross membranes. So in order to understand this better, we have to look at how chemicals are able to cross these lipid bilayer barriers that are present. And so, as you all know, the membranes are made up of lipids, phospholipids, that self-assemble into an aqueous phase on either side of this bilayer, and then in between is a lipid phase. So when we're talking about the ability of the drug to go from outside the cell to inside the cell, or vice versa, you have to talk about how it's able to traverse this bilayer. And how it does it depends on the chemical properties. So when we're talking about a lipophilic molecule, a drug or a xenobiotic that is fat soluble, its ability to cross really has to do with the amount of solubility in lipid. And this is often called the partition coefficient, or log P. And this is just a measure of how soluble it is in fat. And so for lipophilic xenobiotics, or for small molecules, but we'll focus on the lipophilic xenobiotics, they can cross this membrane by simple diffusion. Simple diffusion requires a concentration gradient, so it can only move from high concentrations to low. And with lipophilic xenobiotics, their ability to cross this membrane, again, depends on this lipid partition coefficient. The more lipid soluble it is, the more likely it's going to enter this membrane and be able to cross. But in order to go back into the aqueous membrane, it has to have the optimal aqueous solubility as well, so it doesn't just get stuck in the membrane. Now, if you have a more water-soluble drug or a hydrophilic xenobiotic, then they can't transverse that membrane by simple diffusion. And then we have developed different carrier-mediated or protein-mediated ways that the xenobiotic can cross. And the first one shown here is facilitated transport. Again, this is a specific protein that is recognizing the structure of that xenobiotic and is able to transport it across the membrane. With facilitated transport, again, this is diffusion limited, so it can only go in the direction of the concentration gradient. So it can go in either direction, inward or outward, but it requires a specific binding event to occur at this carrier protein. A similar type of carrier-mediated process for water-soluble drugs are active transport systems that are unidirectional. So these are active pumps that are able to take drugs into the cell or to pump them out. In order to be able to go against a concentration gradient, it requires cellular energy, or ATP. And so this is how we quite often try to import in essential nutrients and try to export out compounds that are toxic or are metabolites that we want to excrete. So that's the basic processes by which they're able to cross membranes. But there's a few nuances to this, and again, this has ramifications for how your drug works from an ADME standpoint. Many of you are familiar with this if you're in the drug discovery space. Looking at a drug and seeing if it's a good drug candidate, you look at these Lipinski's rule of 5, which really just has to do with this idea of hydrophobicity and ability to cross membranes and gain access to cellular components. And some of the important factors here are the size. So if it's greater than 500 Daltons, then it has a difficult time crossing this membrane. If it's too lipophilic, then it's going to dissolve into that membrane, but it's not going to go into the water phase inside the cell. So it's basically just going to get stuck there. If it's too low in lipophilicity, then it's not going to enter that membrane at all, and it would require one of those facilitated transport systems to cross. So simple measures of lipophilicity are very important for understanding this process. But another factor that is often overlooked is polarity. And of course, polarity has a lot to do with lipophilicity, but keep in mind that the charge state of a chemical can change based on pH. And this is because many of our drugs are either weak acids or weak bases. And so depending on the pH, it can change their polarity. And when you're talking about a weak acid or weak base, looking at the standard Henderson-Hasselbalch equation is very informative to say which direction or if your drug is going to be able to cross membranes. What I've shown here is a very simple example where the uncharged drug is the one that's able to cross that membrane because it lacks polarity, and if it has the right lipophilicity, it can cross this membrane. But again, this is driven by polarity and acid-base balance. So if we have a weak acid with a pKa of 4, it's going to be present in a higher percentage in the uncharged state, able to cross the membrane. And now if we go into the plasma where we have a pH of 7, now the uncharged molecule that comes across becomes charged at this higher pH. And so in effect, what we see in this process, just because of acid-base balance, is this weak acid goes from the intestine and is transported across the membrane in a higher percentage at a higher rate than would have been predicted if you don't factor in charge. So one of the reasons why I'm bringing this up is because lipophilic xenobiotics tend to be those that we are the most concerned with from a toxic standpoint. Their kinetics tend to have a lower turnover rate. We have a harder time excreting them. And the question is, why is that? Well, we have evolved ways of getting rid of metabolites and toxic molecules through excretory organs that are water-based. So urine, feces, breath, sweat, even milk, all of these are water-based systems. So if you have a molecule that's lipophilic and doesn't want to move into that water space, then it could be stored, and we'd have a longer half-life, a higher chance for accumulation, and a higher chance for toxicity. So how do we deal with this? Well, what our body is trying to do is take these lipophilic xenobiotics and make them more water soluble so we can excrete them. So how do we do this? We do this through a series of phases, which are often called phase 1, phase 2, and phase 3 metabolism or biotransformation. So this lipophilic xenobiotic will first go through what's called a phase 1 metabolism, which is generally an oxidation step where we're adding polarity, adding some functional or reactive group that is changing the properties of the lipophilic xenobiotic. We're getting a slight change in size and a slight change in water solubility. But as we'll talk about, this is making it a better substrate for the next phase in the reaction. So here's an example with phenacetin, which is metabolized to this 4-hydroxyphenacetin. Again, slight change in size, slight change in water solubility because we've added this polar group. Now, the next step in the metabolism is phase 2 metabolism, which is also known as conjugation. And this greatly decreases the overall reactivity, and it also substantially increases the size and the water solubility. So in this case, our phase 1 metabolite is glucuronidated, or we add the sugar molecule to it. And I think you can see that this greatly increasing the size, the polarity, and the water solubility. Now, the problem is we've kind of created a situation for ourselves where we've taken this lipophilic molecule, made it water soluble on the inside of the cell. So now what we have to do is be able to take this water soluble molecule, have it cross those membranes again so that we can excrete them. And that's where the phase 3 system, or the transport system, comes in, where we use those specific carrier mediated transporters that I talked before. And these often have abbreviations SLCs and ABCs that are looking for this particular cargo, be able to transport it out of the cell and then hopefully be excreted. The two major sites for drug excretion, of course, are through the kidney and the liver, that will go respectively into the urine and to the bile. So again, to continue with this idea of the ADME, but now trying to get a little bit more specific for where this biotransformation is happening, we have to really focus on the liver. And the liver is really the predominant site for drug metabolism, and it's going to be the predominant site for drug-drug interactions as well. And the reason for this is basic physiology. And this is something called the first pass effect. So any type of xenobiotic that is administered orally, like a drug, is going to go through the stomach and go into the intestine, and it's going to cross those membranes, and it's going to be absorbed, and it's going to go into the portal vein and go to the liver. So the first major organ that's seen after the intestine that the xenobiotic sees is the liver. Now the liver is going to take this this drug, and it now can release it into the circulation through the hepatic artery, and now it's going to be distributed throughout the organism, or it can take it and pump it into the bile duct, where it can go back into the intestines and into the feces. So if we look at this in more detail, looking at the liver and looking at how this is arranged, these three vessels that I talked about, the portal vein, hepatic artery, and the bile duct, are really part of a unit called a triad within the liver architecture. And that's shown in this diagram here. So we have the portal vein coming in, bringing the xenobiotic. We have the hepatic artery that's able to transport it into distribution. And then we have the bile duct that is pumping it back into the intestine. And the real workhorse of the liver that is telling the xenobiotic which direction to go is called the hepatocyte. So the hepatocyte is the major cell type of the liver that has a wide variety of different drug metabolism enzymes, a phase 1, phase 2, and phase 3. And now we can create these metabolites, pump them into the hepatic artery to go to the kidney for excretion, pump into the bile duct to go into the feces for excretion. So we're going to be focusing mainly on these liver systems that are involved in drug metabolism. Before we get there, we have to talk about what I think is a fascinating issue for how we try to increase the excretion of particular xenobiotics so that they don't build up and they don't cause toxicity. And of course, here is our metabolic pathway that I've talked about before. And let's take a look at a specific lipophilic xenobiotic, like I showed you with phenacetin before. This goes through a series of reactions that have specific enzymes that are involved in creating the ultimate excreted xenobiotic. So the faster we can do this metabolism, the faster we can get rid of the xenobiotic and decrease its toxicity. So how do we try to facilitate this process? Well, we do this through a series of steps that are basically trying to sense the presence of a xenobiotic. And we have xenobiotic receptors that are responsive to a wide variety of different chemical structures. And then these are going to transcriptionally regulate all of these different enzymes that are involved in the phase 1, phase 2, and phase 3 metabolism. So this adaptive response is talking about how the xenobiotic is inducing its own metabolism. So it's inducing the right enzymes that are going to be able to take this lipophilic xenobiotic and make it more water soluble. As we'll talk about, the xenobiotic receptors, the most important are the nuclear receptors, which are regulating a battery of different phase 1 and phase 2 and phase 3 enzymes. Okay, so let's talk about what this seminar is mainly about, which is the drug-drug interaction. So as we all know, this is becoming an increasing problem, especially in the elderly population, because of the number of different pharmaceutical agents that individuals are taking. Quite frequently, an individual will be taking up to 10 different prescription drugs, and you have to be concerned about how they are interacting with each other within that patient. So there are numerous cases where we know that certain types of drugs cause problems for each other and are able to cause adverse drug reactions through two main mechanisms, as we'll talk about. One is the pharmacodynamic, where one drug is affecting the ability of your main drug, the victim drug, to interact with this molecular target. And another route would be for your drug of interest affecting the victim drug by changing its absorption, distribution, metabolism, and excretion. So we call these pharmacodynamic and pharmacokinetic drug-drug interactions, and we're tipping the balance between efficacy and safety. We're going to spend most of our time talking about this pharmacokinetic end, and we're going to be talking about it on the drug-drug interaction stage. But remember, we're talking about this more from a xenobiotic standpoint. So this could be, yes, a drug-drug interaction, but it could be drug environmental pollutant interaction, drug nutrient reaction, or drug microbiome interaction. Anything that is affecting the ADME of your victim drug is fair game. So, of course, I think all of us have some experience with seeing drug-drug interactions, and all you have to do is look at an insert for a prescription drug, and how many times do you read that you are not supposed to take this medication with grapefruit juice? Well, this is classic drug-drug interaction. And the reason why this is such a common warning label is because a chemical or group of chemicals within grapefruit juice, these coumarins, are inhibitors of a specific enzyme called cytochrome P453A4 or CYP3A4. And this enzyme is the main phase 1 enzyme for 50% of the prescription drugs that are on the market. So 50% of the drugs are using CYP3A4 to metabolize the drug, leading it to higher excretion, and grapefruit juice is going to mess with that ADME balance for the drug. But there are many cases, not just 3A4, any of those enzymes that are part of the phase 1, phase 2, and phase 3 system. This table on the right is showing one particular transporter called ABCB1, which has as its substrates a variety of different drugs that use this for transport. And then we have a series of known the drug-drug interacting inducers and inhibitors that are affecting these particular substrates. And this, again, is for one protein. We have tables and tables of all these different sorts of interactions that can be involved with both drug 1 and drug 2, the perpetrator and the victim. So, we're going to be talking in more detail about how this can be studied, but it's important to think of this as something that is associated with clinical outcomes, but we want to make sure that we identify this early in the process before it gets into the clinic. We don't want this to get out and then have to try to deal with it after the fact. In fact, the earlier we do this, the better. So what we're going to be talking about today is how we can use this to either triage or prioritize drug candidates, identify if your chemical is a potential victim or perpetrator in drug-drug interactions, and hopefully this information will lead to better labeling process where you could have that insert that says, do not take this drug while you're also taking this or while you're drinking grapefruit juice. Also, this will give you ways that if you do go into a clinical study with this particular drug, you're doing it with knowledge and you know what you can start to look for, how you know that you've reached a point where you're starting to see these sorts of interactions. In this talk, we're going to focus on the pharmacokinetic drug-drug interactions, and we're going to focus on nuclear receptors or xenobiotic receptors. And the reason for that is because these are important for a lot of different lipophilic xenobiotics. They're regulators of ADME. And also, we're going to be talking about in vitro approaches. Again, the reason for this is to try to save you time and money, and also you're trying to increase the overall information that you have before you start to go into animal studies or into your clinical studies. Okay, so let's start talking more about the specific pathways and mechanisms, talk about nuclear receptors, and then talk about applications and examples. And first of all, just a really brief introduction to nuclear receptors. This is an area that, of course, we're very much interested in, both in my academic lab and at Indigo Biosciences. These are a family of very important drug targets, but also they're the nutrient targets that are ligand-activated transcription factors. So these are receptors that respond to a variety of different lipophilic molecules. They all have the same basic structure and the same basic way that they regulate cellular activities, but they are distinct. There are 48 separate genes in humans for nuclear receptors, and that's shown in this phylogenetic tree here. But really, they're more similar than they are different, and they all have the same basic structure. And really, the two domains that I want to focus on are the DNA binding domain and the ligand binding domain. So every receptor, and there are a variety of different receptors here, have specific structures of ligand that they're looking for. And so, for example, the androgen receptor is looking for androgen testosterone structure in order to bind and regulate gene expression. So we have specific structures to the ligand binding domain for each particular gene. We also have a similar structure to the DNA binding domain, but they vary in what specific sequence they recognize. So this DNA binding domain is specific for each type of nuclear receptor, and it is looking for a sequence that's found on the genes that it's going to regulate. So the basic mechanism here for all of the different receptors is shown off to the right here, where we have a lipophilic ligand. It's able to cross the membrane, and this is important because these receptors are not found on the cell surface. They're found within the cytosol, or they're found within the nucleus. So they have to get within the space where they can interact with this ligand binding domain. This ligand binding domain responds to the interaction with the drug by changing shape and changing its ability to bind to other proteins. One of the features that I mentioned before is this DNA binding domain. So it is recognizing a specific piece of DNA, and also there are structures that will tell the receptor whether it's going to form a heterodimer or a homodimer. And really, the only reason to bring this up is because it leads to a variety of different types of response elements that these activated receptors will bind to. But the goal here is to have a ligand that is regulating a nuclear receptor, and we have a certain group of genes that are responding to this ligand by a group of target genes that are being expressed. We will also talk about a couple of examples of non-nuclear receptors. They're not of the same family, but they have the same basic mechanism of action, where they have specific ligand binding domains and DNA binding domains, and they have specific genes that they're regulating, such as the aryl hydrocarbon receptor and NRF2. So together, these nuclear receptors and these other non-nuclear receptor soluble receptors, we'll call these xenosensing or xenobiotic receptors, in that they are responding to drugs and other exogenous chemicals to regulate gene expression. To put this back into context of what we're talking about here is, again, we're talking about our adaptive response, and we're talking about our drug being able to interact with these nuclear receptors and driving gene expression. Now, when we are talking about some of these complicated signaling pathways, I always find it's helpful to try to simplify these pathways as much as we can. And we do this through what is often used in environmental studies and other mechanistic types of studies, called an adverse outcome pathway. And so an adverse outcome pathway is shown down here, where we basically put in enough scientific effort where we know the steps that go into an adverse outcome. So we know that there are a certain series of steps that occur. We know the order that they occur in, and we know that which steps are essential between the chemical and the adverse event. And so the way that these adverse outcome pathways are developed is we look at what is the first step that's essential for this pathway. What is the interaction with some cellular component? And this is called a molecular initiating event. This starts the cascade of other responses, leads to a variety of key events that occur, changes within the cell, and all of these changes in cell function culminate in the adverse outcome. So again, if we're thinking about this simplified pathway in the context of drug-drug interactions, what we're looking at here is our drug one victim, where it has a particular pathway that it's going through for absorption, distribution, metabolism, and excretion. But now we have a perpetrator drug that is interfering with this process. And so we'll take a look at this particular pathway and specific examples. But let me just show you diagrammatically what we're talking about here. The molecular initiating event in this adverse outcome pathway is your perpetrator drug interacting with a xenobiotic receptor. That is the first event that occurs. A key event that occurs after this binding, since these are transcription factors, is we get those changes in mRNA that are reflective of changes in activity that is ultimately going to cause the adverse outcome. So these changes are the key events, the cellular events, that ultimately are predicting the change in metabolism, which we're going to call the adverse outcome. So this simplified pathway, we can think of it in a very linear stage that we're showing here, where we have the interaction with the receptor changing the transcription or mRNA, changing the enzyme, drug metabolism, enzyme activity, leading to drug-drug interaction and an adverse drug response. Well, of course, it's not as simple as this. We have a lot of different molecular initiating events and key events that occur. But what we're going to try to do today is try to simplify this. So our goals, of course, are to try to understand if our drug is going to cause drug-drug interaction, have the potential to cause drug-drug interaction in an adverse drug reaction. We want to do this early in the drug discovery process. We don't want to use laboratory animals. So how are we going to do this? Well, we're going to start by talking about molecular initiating events. How do we describe whether a chemical is interacting with these xenobiotic receptors? Then we'll talk briefly about key events, gene expression in a relevant model system. And again, these are predictive of that adverse outcome. So let's start with the approach to looking at molecular initiating events as it pertains to drug-drug interactions. So how we address this is with what's called a cell-based reporter assay. So what we are doing is we're taking advantage of what we know about how these xenobiotic receptors are causing their biological response. We know they bind to ligand. We know that they interact with pieces of DNA and drive gene expression. So the classic example is shown here, where you have the nuclear receptor that is able to bind to a piece of DNA. In this case, we're trying to develop a very simple system to study this. So instead of looking at a target gene, we're introducing an artificial gene that's called a reporter gene. So in this case, if we transfect in the receptor and an artificial gene that is driving a reporter gene, in our case, we use luciferase, the luciferase enzyme. You activate your receptor within this cell. It binds to DNA, recruits the transcriptional machinery to that plasmid, and it drives the expression of luciferase, which you can easily measure. There's another system that we use that takes advantage of the fact that you can move portions of the receptor and they maintain their activity. So this is called a cis-trans assay, where we're able to remove the ligand binding domain, stick it to an artificial DNA binding domain, and then have an artificial DNA binding domain reporter construct. And this has some advantages, as we'll talk about later on, where you can isolate just the ligand binding event that occurs and look at more specific types of responses. So we have built a lot of these different types of reporter assays. These are in cells that are able to respond to a variety of different ligands. All you have to do is plate out your cells, add your test compound, put them back in the incubator, and you could read luciferase activity, and that's going to be reflective of the of whatever the xenobiotic receptor is that's being expressed in that cell. So these are very simple assays. They can be very specific. All of them are robust. You can look at agonist, antagonist, inverse agonist, all within this context. The chemical has to cross membranes in order to interact, so it's more physiologically relevant. And of course, they all are commercially available as kits and services at Indigo. So we'll talk about these types of molecular initiating events as we talk through three types of examples. And we're going to go through the three major drug-drug interaction targets for these particular types of perpetuator drugs. And the most important is PXR, which stands for pregnane X receptor. So this is a receptor that binds to a wide variety of different xenobiotics. So the chemical structures that it recognizes is quite diverse. So it really is a xenobiotic sensing receptor that is basically looking for anything that might be foreign. It'll also interact with endogenous compounds as well, like bile acids and different cholesterol metabolites. But for our purposes, we're thinking about xenobiotics. And again, it's looking for specific ligands. When it recognizes a ligand, it's going to regulate a battery of genes. And this is key. The genes that are regulated by PXR are an alphabet soup of the phase 1, phase 2, and phase 3 drug metabolism enzymes. So the CYP3A, CYP2C family, all of the different transporters that are involved that are coordinately helping get rid of lipophilic xenobiotics. Now I have an interesting example shown here with really how PXR came to prominence. And this has to do with a chemical found in St. John's wort called hyperforin. So what was noticed, and this was some studies that went on in the 90s in Europe, where they were noticing that several women were having pregnancy or the decrease in effectiveness of their birth control pills. And they traced it back to these individuals were taking the dietary supplement St. John's wort. Well, St. John's wort has hyperforin, which is a PXR ligand. Binds to PXR, regulates this whole battery of genes, including CYP3A4. So this is that same enzyme that is inhibited by grapefruit juice. Well, CYP3A4 also metabolizes this progesterone derivative, which is found in birth control pills. So St. John's wort induces CYP3A4, decreasing the effectiveness of birth control pills. Again, classic kind of dietary nutrient drug interaction. So to get back to this idea of how this can be studied, how we can use this. So we can look at potential to induce CYP3A4 by looking at PXR activity. So we can do this by the simple reporter assays that I mentioned before, where you take your test compounds, you add your chemical of interest, and you say, does it increase luciferase activity? So here's an example dose response curve, variety of different chemicals, and you can see you can characterize your compound for the potential to be a perpetrator drug based on efficacy, potency for this response. Now, what you can do as well is you can look at species differences. So we can take these reporter assays and we can design them for specific non-human orthologs. So you can say, well, did my animal study, was that reflective of human drug-drug interactions? And you can do that not by looking at monkey, dog, rat, or mouse. You can look at the PXR constructs from the monkey, dog, rat, and mouse. The next receptor that I'll talk about is one of the most complicated in the nuclear receptor space. It has some similarity to PXR in that it's very important for regulating phase 1, phase 2, and phase 3 enzymes, but it has multiple ways by which it is activated. So we have the classic ligand binding causing a activation and regulating gene expression, but we also have an indirect means of regulating these particular this particular pathway. And really the first activator of CAR that was known as phenobarbital, a very important sedative that we knew were regulating all of these genes, but it's not a ligand for CAR. It turns out that it regulates the activity of CAR through regulating a protein complex, allowing CAR to translocate to the nucleus and regulate gene expression. So you need systems that are able to show both direct agonists as well as indirect agonists in order to look for drug-drug interactions. Now, another thing about this particular receptor that we're finding that there are different varieties of this receptor that are expressed. So these are splice variants, and the splice variants are just slight differences in the structure. So we have the wild type form, we have a splice variant coming from the same gene, the mRNA is displaced. So now we have a 12 base pair insert leading to a CAR2 construct, and then we have a CAR3 construct, which has a 15 base pair insert. Now, what this leads to is differences in the ligand binding domain. And this leads to diversity in the types of ligands that can interact with this ligand binding domain. So we have different agonists for the wild type, the CAR2, and the CAR3. And this is becoming more important in the nuclear receptor space in general. So our assays are able to look at all three different types of ligands that are all going to regulate the drug metabolism, but they're different isoforms. And it's actually a very interesting form of adaptive response where actually ligands like DEHP is probably causing an increase in transcriptome rearrangement to lead to more of the CAR2 receptor relative to the other splice variants. The last receptor that we'll talk about as an example is the aryl hydrocarbon receptor, or AHR. And this was a receptor that was first examined as being a ligand for a variety of different pollutants, such as benzoate, dioxin, PCBs, polychlorinated furans, a variety of again, pollutants that are present. And this is not a nuclear receptor, but it has the same basic mechanism of action: ligand binding to DNA, in this case as a heterodimer with ARNT, regulating phase 1, phase 2, and phase 3, different battery of genes, but again, leading to drug-drug interactions. So different family, same activity. This actually has become a very important drug target. So there are a lot of people that are out there right now looking for HR drug agonists as well as antagonists for looking at a variety of different cases. So again, xenobiotic pollutants now entering into the drug discovery space as well, leading to potential drug-drug interactions with regulation of these drug metabolism enzymes. But I also want to bring up another interesting point with this, in that the aryl hydrocarbon receptor is more than just a CYP1 inducer. It also responds to a variety of different acidic products formed in the gut, including by different microbiome in the gut, that are leading to aryl hydrocarbon receptor activation, yes, leading to CYP1A induction, but also leading to different responses that lead to changes in the GI motility and secretion. So you're actually affecting directly through AHR activation, the absorption at the level of an intestine. So it's a little different type of drug-drug interaction, where now you're affecting the microbiome, the microbiome is affecting drug interactions as well. Okay, so let's shift into how you can look at key events within this particular adverse outcome pathway. So, again, this is something that is commonly done, and we want to look at these key events that are involved in drug-drug interactions. So we want to be able to look at those liver-specific induction of CYP1A1 or effects on CYP1A1, the phase 2 like the UGTs and the transporters. But we want this to be a human system so that we can look at the human metabolism and changes in pharmacokinetics. We want the cells to express all of these drug metabolism enzymes as well as the receptors. So we need a differentiated hepatocyte, and this is often a limitation. We want it to be simple and reproducible and at a low cost. So people that are interested in looking at these key events, they'll quite often pick cell lines because they're easy to look at. These may be, again, human-based things like the hepatoma cell, HepG2, transformed cells like HepaRG, and upsite cells. They may look at primary human hepatocytes, a variety of different 3D model systems that are out there. We focus on what are called upsite hepatocytes, and these are commercially available cells that are transformed human hepatocytes that are derived from a single donor that are easy to expand. This leads to nice reproducibility. They're easy to handle. You handle them just like a cell line, but they express a wide variety of our receptors as well as drug metabolism enzyme. And they have been used quite a bit in the literature to look at drug-drug interactions and different types of altered drug metabolism. So we'd use these cells quite often in a very simplified system, plate the cells, treat them, extract RNA, and then you can look at these key events. So one thing that you can do is extract the RNA and do quantitative PCR, where you're looking for specific drug targets. Shown here is mRNA for CYP1A1, 3A4, and 2B6. So the AHR inducers, the PXR inducers, and the CAR inducers. You can look to see which of your chemicals, if your chemicals are inducing these expressions, this expression. You can look at what happens to metabolism. As you alter metabolism, are you altering cellular function or cytotoxicity? What's shown on the bottom slide here is the ability of aflatoxin to kill these cells. And aflatoxin requires metabolism, CYP3A4 metabolism, to become toxic. And so what we've done here is treat the upsite cells with inhibitors of different phase 1 enzymes, and then treat with aflatoxin B1. And you can see here that the CYP3A4 inhibitor decreases the toxicity of aflatoxin because it decreased the metabolism through CYP3A4. So a very nice system to look at drug-drug interactions. Or you can go to a more holistic approach where you can treat the cells, extract RNA, and then do next-gen sequencing and look at the whole battery of genes that are being regulated. And this is an example of in upsite cells, activate all these different receptors, and you can look at what different phase 1, phase 2, and phase 3 enzymes are being metabolized. So as we look at this and try to understand the pharmacokinetic drug-drug interactions, thinking about molecular initiating events versus the key events, what are the positives and negatives? Both of these assays are relatively high throughput, but certainly these reporter assays are much higher throughput. You can measure hundreds and thousands of chemicals in a single experiment. Easy to perform. Very little cell culture is involved in this, especially with the kits that we provide. Everything is there to run these assays. These can be very specific, down to looking at splice variants, looking at species differences. All of these are positives. Negatives, these are indirect measures. You're just looking at that molecular initiating event. And these assays are built to be robust, to be able to look at this response. We're not really looking at whether you're seeing this activity in a liver cell versus a kidney cell. We're just looking at the potential for this molecular initiating event. Gene expression assays, you can look at multiple targets at one time. So you could look at PXR, CAR, AHR, FXR, all at one time just by looking at the different targets that are being regulated by each of these receptors. You can look at different tissue and species when you're doing these gene expression assays, depending on what cell model system you're using. You get an increase in sensitivity in some regard because you have multiple receptors that are regulating the same drug metabolism enzyme. So you get an additive effect across multiple pathways. And these are considered more physiologically relevant. They're a step closer to the adverse outcome. But they are still indirect because you're not actually measuring the adverse outcome itself, but it is predictive of that. It does require more cell culture as well as other biochemical techniques, gene expression techniques, and certainly lower throughput. Okay, let's try to wrap this up relatively quickly here. So, of course, you wouldn't be at this particular webinar if you didn't think that drug-drug interactions were important. And this certainly is a serious problem in the pharmaceutical and the healthcare industry, especially as our population ages and we're starting to take more and more prescription drugs. So the tools that we talked about are really geared towards perpetrators, but if you know the perpetrator, you can determine whether it's a victim as well. So if you know that it regulates that your particular perpetrator is regulating CYP3A4, you have a whole list of drugs that could be listed as victims for this drug. So this helps you in how you prioritize your drugs, try to minimize the drug-drug interactions by perhaps removing those PXR activators or CAR activators if you're able to from your pool of potential candidates to decrease the potential. You can develop ways to monitor, you know what to look for in the clinical studies and how to label, just because you know what are the particular phase 1, phase 2, and phase 3 enzymes that are being regulated. All of these assays that I mentioned today are commercially available. These are easy for you to perform in your lab for your group of drugs. If you don't have the expertise, we can certainly talk to you about doing this in our service laboratories. With that, I'd like to thank you for your attention, and I'd like to answer any questions that you may have at this time. If you don't have any, you can put them into the chat and we will get back to you.
Fantastic. Well, thanks so much, Jack, for a great presentation. And with that, we'll move right on to the Q&A. All right, so first question here, Jack, is there a specific order of studies to be performed? For example, should reporter assays be done before gene expression or vice versa?
In a lot of cases, this may depend on your level of expertise and what your overall interest is. And really, there is no correct order. So you could first start with the reporter assays to determine if your drug is interacting with PXR or FXR or CAR. And then you could follow that up with gene expression to show that what you saw in the reporter assay is leading to a change as anticipated in the upside cell or the hepatocyte system that you use. And that would confirm the molecular initiating event. You could go the opposite direction where you have data that shows that you're getting alteration in particular drug metabolism enzymes, and you want to know which receptor is responsible, or you want to see if there's species differences. So you may show that, let's say you did a rat study did gene expression and you saw that CYP1A1 was induced, you can then look at the rat aryl hydrocarbon receptor, show that if that is a target, and then you can take that the next step and say, do humans respond similarly to this drug, where we could get that species extrapolation.
Yeah, great point. Next question here. So you said triage assay. What do you mean by triage assay?
That's a good question, and I think that's a kind of a jargon term that I kind of threw out there. And really, triage is just prioritizing to the extreme. So you're trying to get an idea which of your drug candidates should you put forward into the animal studies. And you may decide that there is kind of a line that you don't want to cross. And let's say that you don't want to see PXR activation, or you don't want to see CAR activation because it's going to cause problems with some other drugs through drug-drug interaction. So that would be prioritizing to such an extent that you are removing drug candidates from going forward at all.
Excellent. Yeah, great answer. And I think in the interest of time, we'll just make this next question the last one. They say, let's say my drug activates PXR. How would that help me in determining what other drugs may be impacted? I think you sort of touched on this a bit at the end, but maybe you could just clarify.
Yeah, I think one thing to keep in mind is that there are a lot of resources out there that will tell you which drugs are metabolized by a variety of different specific phase 1, phase 2, and phase 3 enzymes. So if, for example, I mentioned CYP3A4, we have a long list of all of the different pharmaceuticals that are metabolized by CYP3A4. We also know that that enzyme is regulated by PXR. So by inference, if you activate PXR, that whole list of chemicals that are CYP3A4 substrates are going to be affected. And you could do this with each of the different nuclear receptors. You could look at all the different lists of different enzymes. So you're really you're taking advantage of all the resources, all the information that we have on some of the more typical prescription drugs that are on the market.
Yeah, great point. All right. Well, Jack, thanks so much for joining us today, and thanks so much for your insights today. It's really been a pleasure having you with us.
You're welcome. Thank you.
Yeah, our pleasure. Also, big thanks to the audience for joining us. And in closing, we hope you enjoyed this Scientist.com Inside Scientific and Indigo Biosciences webinar, and we'll see you again next time. Have a great day, everyone.